Good evening, guys, and today is our last lecture of glycolysis. Finally, we have covered all the processes and important steps of glycolysis. So let's in this lecture let's revise a little bit and conclude our topic. First, glucose enters into the cell via the transporters is converted into glucose 6-phosphate. It is a trapped form of glucose, okay? And this step is the first irreversible step. It is using the enzymes hexokinase and glucokinase. Hexokinase is present in most of the tissues of our body, and glucokinase is present mainly in the liver, and this is the first step where the ATP is invested. Okay, this is the investing phase of glycolysis. Glucose 6 phosphate is reversibly converted to fructose 6 phosphate by an isomerase enzyme because these both structures are biologically same. Fructose 6 phosphate is then converted to fructose 1,6 bisphosphate. It's the second irreversible step of glycolysis. Here the second ATP is invested. This step occurs via the using of enzyme phosphofructokinase 1 or PFK1. PFK1 is a very important regulated enzyme. Its positive regulators are fructose 2,6 bisphosphate, ATP, and citrate. I have talked about these in previous lectures. And its negative regulator is AMP and is also affected by hormones insulin and glucagon. Okay. The next step is the conversion of fructose 1,6 bisphosphate to one molecule of glyceraldehyde 3-phosphate and one molecule of dihydroxyacetone phosphate. But dihydroxyacetone phosphate must be converted to glyceraldehyde 3-phosphate before it goes into the subsequent steps of glycolysis. So dihydroxyacetone phosphate converts into glyceraldehyde 3-phosphate, and now we have two molecules of G3P. I will abbreviate that, and this step is occurring via the help of enzyme aldolase. Now comes the yielding phase of glycolysis. Now we are going to yield ATPs from the ATPs we have invested here, right? It's like a business. So G3P is converted into 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate by the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. Why dehydrogenase? Because we are converting two NAD positive molecules into two NADH molecules, and this NADH is very important because this is the reservoir of energy. It will go to electron transport chain and mitochondria. It will yield us how many ATPs? It will yield us 3 ATP per NADH molecule. It means that we have 2 NADH molecules. So how many ATPs? 6 ATPs. Okay. After that, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate by phosphoglycerate kinase, then to 2-phosphoglycerate by enolase. Okay, 3-phosphoglycerate to 2-phosphoglycerate by phosphoglycerate mutase. After that, the 2-phosphoglycerate is converted to phosphoenol pyruvate, which is known as PEP, by the enzyme enolase. And this is the last irreversible step of glycolysis, in which the PEP, i.e. the phosphoenol pyruvate, is converted into the pyruvate. Two molecules of pyruvate are obtained from one molecule of glucose. And in which steps we are in yielding the ATPs? Via the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, and by the conversion of PEP into the pyruvate, we are yielding two molecules of ATP there and two molecules of ATP here. So we have in general 4 ATP yielded in glycolysis, but we have invested 2, so net gain of ATP in aerobic glycolysis is 2 ATPs. But when we combine them with the ATPs we will obtain from NADH, so 6 plus 2 is 8 ATPs in general we obtained from aerobic glycolysis. But in anaerobic glycolysis, when there is no mitochondria, we will only have 2 ATPs. Regulation of pyruvate kinase. Pyruvate kinase is this enzyme, and pyruvate kinase converts PEP into the pyruvate, and pyruvate kinase is regulated by hormones like insulin and glucagon. When we are in high fat state, it means that our blood glucose level is high, So insulin is high and glucagon is low. When insulin is high, it is the positive regulator of pyruvate kinase. It indicates the pyruvate kinase to proceed the glycolysis forward. When we are hungry, like we haven't eaten in 5 hours or 8 hours, our glucose level is low, our insulin is low, and glucagon is high. Okay, when glucose is gone, then glucagon will be there. And glucagon, via the series of steps, via the G stimulatory, then it stimulates adenylate cyclase, CAMP goes up regulated, then protein kinase A. I have discussed all these steps in the previous videos. Protein kinase A will phosphorylate the pyruvate kinase, and it is the negative regulator of pyruvate kinase. It will stop the glycolysis. It will inhibit the glycolysis. Think logically that when we are hungry, do we need to process the glycolysis and we need to obtain energy from glycolysis? No, we are not utilizing that glucose which is already present in our cells. But when the glucose level is high, when we are in fed state, then yes, then we can proceed the glycolysis. So it's the positive regulator. Talking about the red blood cells rely only on anaerobic glycolysis because these cells lack mitochondria and electron transport chain is not functional. When electron transport chain is not functional, so NADH obtained from glycolysis cannot go to mitochondria and cannot yield us 6 ATPs that were yielding in aerobic cells. So net gain of ATP in anaerobic glycolysis will be only 2. Okay, but in other cells in which mitochondria is functional, NADH obviously goes to mitochondria and electron transport chain and will yield us 6 ATPs, 3 ATP per NADH molecule. But in RBCs, how we are going to regenerate NAD positive? It's very important molecule NAD positive. Here you can see that this NAD positive is converted into NADH, right? But for the process of glycolysis to be going on, we need like bulk of NAD positive. So in aerobic cells, in the cells those are performing aerobic glycolysis, their oxidative phosphorylation is working. In electron transport chain, when the NADH is processed, it will convert back into the NAD positive. It means in aerobic cells we will regenerate NAD from the from the electron transport chain. But in anaerobic cells where there is no electron transport chain, how we are going to regenerate NAD positive? Nature has set up a very important mechanism that in anaerobic cells, pyruvate is converted to lactate by lactate dehydrogenase enzyme, and in doing so, NADH is converted to NAD positive, and here we go. We have regenerated NAD positive. That's what we needed for the glycolysis to be proceed. But in case of anaerobic glycolysis, NADH is used for the conversion of pyruvate to lactate, and we have obtained NAD positive, so there is no net generation of NADH. Okay, and in anaerobic glycolysis 2 ATP were being produced, and these 2 ATPs are enough to meet the demands of RBCs because RBCs are metabolically inactive cells. They only just transport the oxygen and hemoglobin. The next important topic is the pyruvate kinase deficiency. We are talking about this enzyme. Okay. Pyruvate kinase deficiency can result in anemia. How it occurs? Let's talk about some pathophysiological mechanisms. That pyruvate kinase is used to convert phosphoenol pyruvate to pyruvate and yield us 2 ATP molecules in this step. But if the gene of the pyruvate kinase is mutated, the protein will be misfolded or protein will be abnormal. The enzyme will be abnormal. This enzyme will have reduced affinity for its substrate. What is the substrate for pyruvate kinase? Phosphoenol pyruvate, right? It will have reduced affinity for PEP. It will have reduced reaction velocity, which is Vmax, and glycolysis will be decreased. Its speed would be decreased and ATP would be decreased. Okay, in this step. But when ATP is reduced, it has very negative impacts on the RBCs, how it occurs. Let's talk about where the gene is actually mutated. As you know that RBCs, the mature RBCs lack nuclei. They do not have nuclei. Okay, so the gene cannot be present in the mature RBCs, but they can be present in RBC precursors. What are RBCs precursors? These are erythroblast. These are erythroblast going through a series of reactions and is converted into reticulocytes, and eventually we got the erythrocytes, which are RBCs. In erythroblast there is genes of pyruvate kinase, and here they are mutated and they are not functional when the RBCs get mature. So we were talking that ATP become reduced. As you know that all cells of the body and including the RBCs, they have special pumps known as sodium potassium pumps in their membranes, which are constantly working. They expel sodium out and retain the potassium in the cell. They export 3 sodium out and they retain 2 potassium into the cell. And these pumps are very important for the maintenance of the biconcave normal integrity of the RBCs. If these pumps are not functional, okay, when there is low ATP, these pumps are like active transporters. So when there is less ATP, these pumps cannot work. The ATPases cannot work, and sodium accumulation inside the cell starts to occur, and sodium attracts water and the RBCs become swell and they lose their biconcave integrity and they become spherical. Okay, that's here I have written that sodium potassium pump become dysfunction, sodium accumulates inside the cell and water follows, it results in the cellular swelling. These spherical RBCs, they are less flexible and they cannot pass through the narrow capillaries which are present in the spleen. As you know that the full lifetime for the RBCs is 120 days. After that, they are degenerated by the spleen. But these RBCs, what we are talking about, that these are not like a mature RBC, they are premature. They have not completed their 120 days yet, but when they pass through the spleen, there are special cords which are known as cords of Billroth, which are known as the cords of Billroth. Here they are trapped because these cords have very narrow size and the RBCs are trapped here. The macrophages present in this area recognizes these RBCs as pathogens and destroy them prematurely because they have not completed their 120 days yet. So they are destroyed prematurely and it will result in hemolytic anemia. Now you understand that how pyruvate kinase deficiency results in hemolytic anemia. It will cause the ATP dysfunction, the sodium potassium pump ATPase dysfunction. Sodium accumulate inside the cell and water follows. The RBC swells up and they are destroyed in the cords of Billroth which is present in the spleen. And why it's called hemolytic? Because there is hemolysis going on, and anemia, reduced hemoglobin levels. Eventually that RBC's level goes down. Also with RBC's hemoglobin level goes down because hemoglobin is present in the RBCs. The second enzyme deficiency is known as the glucose 6-phosphate dehydrogenase deficiency. In the conversion of glucose to glucose 6-phosphate, after that it must be converted to fructose 6-phosphate, and here the glycolysis is going on. But this enzyme known as glucose 6-phosphate dehydrogenase, it converts the glucose 6-phosphate and diverts it into the pentose phosphate pathway. This pathway we'll discuss in another lectures, but you just remember that glucose 6-phosphate dehydrogenase, it will divert the glucose 6-phosphate towards the pentose phosphate pathway. And when this enzyme is deficient, it will impair ATP production due to disruption of glycolysis. Okay, the glycolysis is not going on and ATP production would be less. Talking in general about anaerobic glycolysis, that it occurs in those cells that lacks mitochondria, for example RBCs, renal medulla, testes, lens of eye, and the tissues with severe oxygen deficiency. These cells lack the mitochondria So they are dependent on the anaerobic glycolysis. But you just remember that cells with mitochondria, but when they have hypoxia, when oxygen is low, the oxidative phosphorylation cannot occur. Okay, oxidative phosphorylation cannot occur, and NADH, which was produced in the glycolysis, cannot be utilized when there is hypoxia, when there is no oxygen. So this NADH will start to accumulate inside the cell, and this NADH is a trigger. It's like a tag or trigger that is used to convert pyruvate to lactate. It will regenerate NAD positive from there, as we have discussed that in the conversion of pyruvate to lactate, NADH will goes to NAD positive and we are regenerating NAD positive. When there is NADH accumulation, anaerobic glycolysis starts. This is the rule. Okay, energy metabolism during the exercise. When there is exercise going on, it's not like a normal exercise, it's like a strenuous exercise. The oxygen supply will be low, and oxidative phosphorylation is not working because oxygen is low. So NADH will start to accumulate inside the cell, as I've talked about it, that this is the rule. When NADH starts to accumulate inside the cell, it will shift the glycolysis from aerobic to anaerobic. It's very important. In anaerobic glycolysis, lactate dehydrogenase converts pyruvate to lactate, and in this step we are regenerating NAD positive. Like same things are repeating here. But one thing new is that when there is lactate build-up, it will cause muscle cramps because of the acidosis. The lactic is an acid, right? So it starts to increase in our bloodstream and it will cause acidosis in the body. Anaerobic glycolysis yields 2 ATP, but there is no net production of NADH. Why there is no net production of NADH? Because that NADH is used for regeneration of NAD positive. So net production of NADH would be no, zero. There is some difference between aerobic and anaerobic glycolysis. First of all, location. Aerobic will have cytoplasm as well as mitochondria, but anaerobic is only going to occur in the cytoplasm because there is no involvement of mitochondria. Oxygen requirement, yes, aerobic glycolysis will have oxygen, but anaerobic will be independent of oxygen. ATP yield, as I've told you that 2 ATPs are produced in anaerobic glycolysis and 8 ATPs are produced in aerobic glycolysis, including the ATPs obtained from the oxidative phosphorylation. End products in aerobic, 2 pyruvate molecules are there plus 8 ATPs. In anaerobic, there will be 2 ATPs plus lactate because pyruvate was converting into the lactate. Process continuation. NAD positive will be regenerated from the conversion of pyruvate to lactate. Yes, we have discussed that. But in aerobic, NAD positive will be regenerated by the electron transport chain. I have told you that. Relevance of RBCs, that RBCs are solely dependent on the anaerobic glycolysis, but RBCs are not performing aerobic glycolysis. In case of severe hypoxia, hypoxia can occur in which conditions? Let's discuss about that. First of all, if there is a vasal obstruction, yes, the blood cannot be supplied to a particular tissue, there will be hypoxia. Generalized hypoxia or severe hypoxia. If there is heart attack or circulatory shock, yes, there will be hypoxia and ischemia. Loss of blood from the body, it will cause reduction of volume of the blood and it will eventually leads to generalized hypoxia. Pulmonary embolism, in which the blood is not being oxygenated by the lungs, yes, it will cause ischemia and hypoxia. Strenuous exercise and heavy sweating, it will also result in hypoxia. And when there is hypoxia, as I have told, that oxidative phosphorylation cannot function and it will cause NADH accumulation inside the cell. Okay, and this NADH accumulation, there is a rule that it will switch aerobic to anaerobic glycolysis. And in anaerobic glycolysis, we are yielding 2 ATPs per glucose molecule, and this 2 ATP is enough to sustain the cell temporarily. Okay, when there is hypoxia, we are not performing oxidative phosphorylation, but at least we are performing anaerobic glycolysis to sustain the cell temporarily. But the side effect is that lactate will accumulate inside the body. It will cause lactic acidosis. There is also other term that if hypoxia persists or glycolysis is inhibited by toxins, it can cause energy failure and cell death. What about lactate metabolism? We are producing lactate in so much amount in anaerobic glycolysis. So what to do with that lactate? How this lactate is going to perform in our body, and which tissues are utilizing this lactate? The conversion of pyruvate to lactate, it will regenerate NAD positive. I have told you thousands of times that. So lactate is utilized by two primary tissues. First is the heart and second is the liver. The heart will convert lactate back to the pyruvate. Okay, lactate back to pyruvate. So when we were converting pyruvate to lactate, we were using NADH, but in conversion of lactate back to pyruvate, we are using NAD positive, and this pyruvate will enter the Krebs cycle or citric acid cycle and will yield us a lot of ATP. In liver, lactate is converted into pyruvate, same as in the heart, and this pyruvate can either enter the Krebs cycle or citric acid cycle, or it can participate in the gluconeogenesis. We'll talk about that later, how gluconeogenesis is going to perform. The last topic for today's discussion, and hopefully the end of glycolysis series, is the alternative fates of pyruvate. Now we have pyruvate. What are the fates of that pyruvate? In which mechanisms or in which steps this pyruvate is going to participate or perform? First, when there is no oxygen, mitochondria is not there, so it will convert into lactate via the lactate dehydrogenase, and it will regenerate NAD positive. We are talking about this in the whole lecture. The second is that when the oxygen is present and mitochondria is present, the oxidative decarboxylation of the pyruvate is going to go via the enzyme pyruvate dehydrogenase, and it will convert into the acetyl CoA. It's like a general step that most of the textbook are talking about, that pyruvate is converted to acetyl CoA, and this acetyl CoA will goes into the Krebs cycle and yield us a lot of ATP, right? So this step occurs via the help of enzyme pyruvate dehydrogenase. It is a decarboxylation mechanism because here carbon dioxide is being removed. Because pyruvate is a 3-carbon molecule and acetyl CoA is a 2-carbon molecule, where one carbon goes? Is it vanishing or is it disappeared? No. Carbon dioxide is formed in this step. The third step is that pyruvate can also form one of the intermediate of the Krebs cycle known as the oxaloacetate. And oxaloacetate is a 4-carbon molecule from where one carbon comes. It will come through the carbon dioxide via the utilization of carbon dioxide. Pyruvate combines with carbon dioxide and will form oxaloacetate. Next fate of pyruvate is the conversion into the fatty acid. When we are in high fat state, a lot of pyruvate is there, a lot of ATP is there, and excess of pyruvate is directed towards the fatty acid synthesis. Okay, it will convert into the fatty acid synthesis. We'll discuss about that later, but this fatty acid is going to make us fat. The last fate of pyruvate, it's not occurring in humans but occurs in the microbes. It is the conversion of pyruvate to alcohol, which is ethanol, via the addition or via the help of acetaldehyde. And these 5 are the fates of pyruvate. These are alternative fates. Either of them can happen according to the body's need and depending on the situation of our body. So this was for today. I hope you understand the lecture, and this was for all the glycolysis thing, and you can watch the previous lectures as well to clear your mind on the biochemical steps of glycolysis. So I will see you in the next video talking about another topic. Until then, goodbye.